very happy that you're all able to join us this evening for tonight's presentation, Pediatric Epilepsy Disorders. And before I introduce Dr. Mirzaa, just wanted to go over a couple different housekeeping issues. First of all, everyone will be receiving an evaluation link towards the end of this program. And I know Dr. Mirzaa would really appreciate it if you could just take a couple minutes and give feedback on tonight's presentation. As well, if you have a question during the presentation, please feel free to put it in the chat and I will interrupt Dr. Mirzaa and ask. And then at the end, we will unmute you and you can ask questions live. And I know she would really appreciate a two-way dialogue, so feel free to do that. And with that said, Dr. Mirzaa is a geneticist and researcher with board certifications in pediatrics from the American Academy of Pediatrics and Clinical and Molecular Genetics from the American Board of Medical Genetics and Genomics, excuse me. Dr. Mirzaa graduated from the Arabian Gulf University in Bahrain and then pursued pediatric residency at the University of Illinois in Chicago, followed by fellowships in clinical genetics and molecular genetics at the University of Chicago and the University of Washington. She is currently an attending at Seattle Children's Hospital, who staffs the Epilepsy Genetics and Neurogenetics Clinic, program director of the Neurotherapeutic Clinic, and the co-director of the Molecular Diagnostic Lab at the University of Washington. Her research focuses on many aspects of human developmental disorders, including understanding the causes, mechanisms, and outcomes of these disorders and associated comorbidities, including autism, epilepsy, and intellectual disability, with a special focus on brain malformations. So with that, I would like to hand over the time to Dr. Mirzaa, and thank you again.
Thanks, Patty. And thank you for inviting me to present this evening. It's a pleasure to be here and talk to you all about a topic that is near and dear to my heart and a rewarding journey for sure. As Patty mentioned, I would absolutely welcome this being, hopefully an engaging kind of time. So if anybody has any questions, comments, please feel free to interrupt and ask. I think it would be super fun for us to chat. So the title of my talk is really on pediatric epilepsy disorders, which is something that when I came here from Chicago now 10 years ago, was a program that, it's a longstanding program. We're one of the largest epilepsy centers in the country. Certainly our neurology and neurosurgery expertise are, world-class. And on the genetics side, I was privileged to be among the few geneticists who were involved in starting the Pediatric Epilepsy Genetics Clinic and really trying to figure out what is the best way we can support and diagnose children with early onset epilepsy, and especially the severe subtypes that require sometimes epilepsy surgery. So with that, I'll kind of jump in here. And so as an outline, I thought I'd just kind of start very briefly about talking about why should we even, genetically study pediatric epilepsy disorders to begin with. And apologies if some of this might be preaching to the choir, really. But then I think another kind of quick point that I thought I'd touch base about is, what is the value of a genetic diagnosis, right? Why is it important, and why should we even pursue it in the first place? And there's a lot of facets to that, I think, sometimes that we might not appreciate, on a regular basis. And then I'll kind of bring up examples throughout this talk. Really, I'll focus on an example of a genetic pediatric epilepsy group of disorders that are, very fascinating and really, I think, highlight a lot of like the practical kind of takeaways that I hope you will find useful. And again, happy to really chat and answer questions. So in terms of pediatric epilepsy disorders, again, this is really preaching to the choir to some extent. They're relatively common disorders. So, 65 million people worldwide are assessed to live with epilepsy. 3.4 million in the United States live with epilepsy. And as we all know, epilepsy often does not, exist in isolation. For many, particularly for early onset pediatric, epilepsy syndromes, epilepsy is often one of many comorbidities that a lot of these children have to live with, and include, of course, cognitive deficits, autism or autistic features, sometimes even sleep disorders, psychological problems such as depression and anxiety, migraines, ADHD. There's a lot more that's not on here as well, such as feeding difficulties, sensory disturbances, so on and so forth. So it's a really complex, often complex set of symptoms that the families have to live with. And when it comes to genetic syndromes that are associated with epilepsy, it's a really large number. So, in OMIM, there's apparently more than 500 syndromic associations where epilepsy is either a key feature or an associated feature. And in spite of all of this wealth of knowledge we have about how common it is and how severe it can be, the diagnosis and oftentimes medical management can be non-uniform. And I'm speaking specifically to the genetic subtypes. So we still have a bit of ways to go to improve sometimes the journey that the families have to go through on the diagnostic side. Now, I like to kind of link where we come from in genetics in terms of genetic technologies and genetic testing. I kind of like to think about it in a way that's similar to how perhaps neuroimaging kind of has come along. If you think about it, in the 80s, for example, or in the 70s and earlier on, we really didn't have, and this is actually a typo on my slide. We didn't have, these are not MRI images, these are actually CT scans. So we had CT scans basically to kind of allow us, for the most part, look at the morphology of the brain for kids who had early onset epilepsy and see if there's any structural abnormalities. And this is the structural brain malformations have been an area of high interest to us here. So, and based on the resolution of the technology at the time, we had an idea of several subtypes and several morphological abnormalities such as lissencephaly, polymicrogyria, heterotopia, maybe FCD if it's severe enough to be within the resolution of a CT scan. Fast forward, of course, now we have much higher resolution neuroimaging, right? And our MRI qualities keep getting better and better. And so we actually have a much higher understanding of the range of morphologic abnormalities in the brain that can occur. And the degree of, it's always fascinating to look at brain MRIs of children with our neuroradiology colleagues, because you can see just a fascinating spectrum of structural abnormalities on these really, really terrific quality MRI scans. So in a way, genetic testing has kind of come along a similar path, where the quality of it just kept getting better. The resolution is much better. So our ability to look at our DNA at a much closer level has only improved dramatically. So if you, to age myself here, really, for some of us, as we were training, for example, we didn't have a lot of the sophisticated genetic testing we have today. We would start, a lot of the time, the testing strategy by looking at chromosomes. Sometimes we do metabolic testing, and then we kind of start with single gene testing. And for many families, even just doing these tests can take years, right? Especially if we do one gene after another. Now, of course, we have much better genetic modalities, genetic testing modalities that are collectively called next generation sequencing methods that include, multi-gene panel testing, especially for epilepsy, exome sequencing, and now genome sequencing, right? And so it's very similar to kind of what other diagnostic modalities have gone through. And we're very lucky to really have the tools we have today. And this is actually a really nice figure that I like from a paper from a few years ago that showed really how important these technologies were in advancing our understanding of the genetics of epilepsy, for example. So you have the channelopathy era, which was kind of in the mid-90s to the early 2000s, and this was prior to these advanced tools, prior to NGS, where, we kind of were identifying new epilepsy genes, albeit at a slower rate. Most of these were channelopathy genes. Then when NGS came along, and this is shown over here in the red, you can see it rapidly accelerated gene discovery. And it just kept shooting up. It kept shooting up from there, and this is even right at the beginning of when exomes were starting to be done widely. Now we're starting to do genomes. So, I think this trajectory is probably going to continue to accelerate. So with that, I just wanted to quickly kind of mention, just so we kind of all feel comfortable with these terms, is when we talk about next generation sequencing, which are, again, the newer genetic tools we have, there's three general flavors, if you will, of these tools. The first one is what's called targeted sequencing, which means that you have, for example, a specific differential diagnosis where you suspect a certain number of genes to be involved. So you'd like to kind of really test those genes. A great example is epilepsy panels. So there's a lot of multi-gene panels that are targeted for epilepsy that really include a lot of the epilepsy genes. So these are targeted to coding regions within those genes. Then we have exome sequencing. So exome sequencing is much broader than a multi-gene panel or targeted sequencing, and it looks at all the coding regions. So this is what's shown in the blocks here. These are the coding regions of the genome, and this is what in red are, of course, it's the coverage of that test. So this is what exome sequencing typically covers. And now, of course, we have whole genome sequencing, and whole genome sequencing looks at all of the DNA. So coding, non-coding, the entirety of our DNA as shown here. That is what whole genome sequencing does. Now, of course, as a general rule, the broader you go, the deeper the less deep you can be. So meaning that if you do genome sequencing, you're probably not going to be able to sequence very deeply certain regions of the genome, because again, your compromise there is you want to look at broad regions of the DNA, but any one region you cannot really go deep into it. And I'll kind of talk about what that means in a second. When we talk about mosaic brain disorders. So as a kind of a final really takeaway for the differences between these modalities, we used like this apple analogy many years ago, and this is from the UW Genome Sciences Center, this figure, where it shows basically the exome is really kind of taking a slice of the apple, which is just the protein coding portion. A targeted panel is almost like you're looking at cores within the apple itself. You look at specific regions within that slice. So that's just the protein coding portion. The genome looks at the entirety of the apple. So that's really what it is. And just actually last week, we have been approved to offer whole genome sequencing for children seen in the epilepsy genetics clinic. So this is really, really relevant to us right now. And the value of this has been really impactful, because if you think about brain malformations, for example, a lot of these are associated with early onset epilepsy. And here I'm showing kind of broad categories of different brain malformations, ranging from migration disorders to abnormalities in proliferation, or microcephaly, so growth problems such as microcephaly or macrocephaly. So if you look across all of these, what next generation sequencing has really done is the following. It really allowed us to identify so many genes, and more importantly, the networks and relationships between these genes, that has been really revolutionary. And in a way, it's kind of like the journey that oncology has gone through, because initially it was very kind of like slow discovery, and then we started to understand all these cancer pathways. The same thing has happened in neurosciences and brain malformation research. And the value of this is that when we think about these cellular networks, we can perhaps start to think about therapeutic targets for these pathways or networks. So it's really, really valuable to uncover all of these intricate networks and relationships within the cells. So this is really where we're at right now. We've gone through this journey over more than a decade. And certainly this has been throughout my career now. And we're finally at the stage of doing whole genome sequencing. Great. So does that mean we're done? Is it all smooth sailing from here? The reality is no. So sadly, we have significant diagnostic challenges. We've come a long way, but there's still really true issues that we need to kind of address. And without really talking about them, we really can't address them effectively. So some of the diagnostic challenges that still exist is that most epilepsy disorders, including pediatric surgical epilepsy disorders, are often severe. And there are several, so it's not one disorder, right? So you can't really have one testing strategy for everything. And they're associated with a lot of comorbidities, as we talked about. So this creates a lot of challenges, because a lot of these families go through so much, and their children go through a lot of, medical care, that to add genetics to it is adding an additional medical evaluation that is often complex, it's often scary. So we really need to be, aware of that. Importantly, when it comes to, making the case for testing, whether it's with insurance or with funding agencies or things like that, sadly, there's a misconception that these disorders are all rare, right? So these are all rare. Why should we test them? These are isolated incidents. Why should we test? So the third kind of diagnostic challenge is that access to genetic care is a true challenge still. Our wait lists are really long. We have a shortage of geneticists across the country and across the world. So it takes a long time sometimes to be able to get to a geneticist, and sometimes it can be years. And we're privileged here that we have one of the larger genetic teams in the country. Other places in the country don't, not as much. And then lastly, we have genuine barriers toward genetic testing, whether it's, again, insurance coverage or others, or, the level of coordination, the infrastructure, that you need to coordinate such complex testing is really high. It's very involved testing. So you really need a lot of things to line up in order to get it done. So really, these are some of the diagnostic challenges we have. Now, with that, I really want to share with you, start to share with you this journey we've gone through with a very important and interesting subset of pediatric epilepsy surgical disorders, which are mosaic disorders. And this is my excuse, by the way, to start showing these artistic images of mosaic pieces of art that I absolutely am obsessed with. So this is, and of course, Pacific Northwest style. It has mountains. So I really wanted to share with you some of the examples of these disorders and highlight some of the salient concepts that could be helpful. So when I was a medical student, I thought I'd kind of share this example of a syndrome that's really important from a conceptual way. So as a medical student, I was lucky at the time to work at the University of Chicago and do a summer, research project. So it wasn't snowy at the time, even though Chicago, you can argue how the weather is bad year round. But so there was a summer project that we had, and this was actually a project that involved Dr. Ian Glass, who is one of our longstanding geneticists here at Seattle Children's, for those of you who may be familiar with him. But we had basically a group of old paper charts of these 5 or 6 kids who had a very unique combination of features that we felt like was a syndrome on its own. And these kids had brain overgrowth or megalencephaly, polydactyly, postaxial polydactyly, which means extra fingers, polymicrogyria, which you can see over here, and hydrocephalus. A subset of them had hydrocephalus. You can see a little bit of ventriculomegaly over here. So we decided to describe this syndrome, and we called it MPPH syndrome after the 4 key features of this syndrome. And we published it. This was more than 10 years ago, more than 20 years ago. Oh my gosh, I'm completely old. And that was that. And so we thought then at the time, this was, a syndrome that was newly described. We didn't know the genetics of it. Fast forward, after I finished my training, I really wanted to come to Seattle in part because of the dynamic scientific community here. So, a lot of the next generation sequencing tools, needless to say, were actually developed here in Seattle, both at the Genome Sciences Center, and then SCRI adapted them quite quickly. And so a lot of NGS, especially exome sequencing, was launched here. So this was part of the reasons why I decided to come here at the time, and we decided to try to test some of the samples of children with MPPH syndrome by exome sequencing. And lo and behold, we've identified 3 causal genes for this syndrome: PIK3R2, cyclin D2, and AKT3. These 3 genes lie within an important pathway known as the MTOR pathway. And so this is the pathway right here. This is just a schematic of the pathway. It's a highly complex pathway, as many of you who probably have, it's such a prolific pathway that a lot of people have worked into MTOR space, but it's an important cancer pathway as well. And our group, as well as several others across the country and the world, were doing kind of similar, using these technologies in a similar fashion. So we identified not only these 3 genes, but many other genes that I'm showing here in a wide range of brain malformation syndromes. So there's a whole spectrum that I'm showing here at the upper edge of the slide that were all found to be associated by mutations of this pathway. And the genetic differences between them were interesting. So some of these mutations were gain of function mutations of activators of the pathway, or loss of function mutations of inhibitors of the pathway. And what that means is that essentially this pathway was hyperactivated in all of these. So it's almost as if you're either pressing on the gas pedal or taking away the brake of a car. The endpoint was the same, is that it was activation, abnormal activation of this pathway, similar to what has been described in cancer. And that was actually really important because this pathway is overactive, and this pathway has been highly targeted by pharmaceutical companies and other therapeutic avenues for cancer therapeutics. There's a whole range of inhibitors in the market, including the infamous, of course, sirolimus or everolimus or rapamycin. And then there was a newer drug that I'll talk about later on that just came out, a new inhibitor that just came out for PIK3CA positive breast cancer in women just a few years ago. So this kind of put us in a very interesting spot about where we thought maybe we could start thinking about therapeutics for these early onset epilepsy disorders using the same strategy. So we found again that this was a big spectrum. Again, it ranged from really very focal forms of cortical dysplasia that are caused by mutations of this pathway, all the way to diffuse brain overgrowth with abnormalities, diffuse abnormalities, in addition to, of course, the classic hemimegalencephaly, where one half of the brain is involved. All of these were now found to be caused by mutations of the MTOR pathway, which is very interesting. So this is the pathway again, just to kind of show it. And I'll show you like a few quick examples of what these disorders look like. So mutations, for example, in a gene upstream of the pathway known as PIK3CA are associated with the entirety of the brain spectrum that I just showed you, anywhere from FCD to hemimeg to megalencephaly. Similarly, mutations of PIK3CA are actually associated with the same kind of variability in the body. So it can be associated with isolated overgrowth, with macrodactyly, to limb overgrowth, with vascular malformations, to really severe and pervasive forms of overgrowth, such as something called Cloves syndrome. All of these disorders, with this broad range, were found to be caused by mutations of the same gene within the mTOR network. And interestingly, the mechanism was a little bit different. When we think about how do genetic mutations happen, generally speaking, most of them are what we call germline mutations. What that means is that the mutation arises early on during development, during fetal development. It can arise in a sperm cell, for example, or an egg cell, and then propagate to the entirety of the fetal tissues and organs as they're developing. So this is the most common way for genetic disorders to occur. And the fetus would, by definition, then have the mutation in all tissues, in all cells, equally. So it would be what's called a germline mutation. For mTOR pathway related disorders, it's a different mechanism. Many of these, not all, are post-zygotic, which means that the mutation arises a little bit later on during fetal development, potentially at the 2-cell stage or 4-cell stage, or so on and so forth. And as a result, because it occurs later on during fetal development, it's not present in all tissues of the fetus. It's present in some tissues, some cells. So if hypothetically it occurred in a brain in an early neuroprogenitor cell, then in that case we have a mosaic brain malformation. And similarly, it can occur, of course, in the body as well. So this is what a mosaic mutation is, and this is the most common for mTOR-related disorders. And in a way, if you think about it, again, this is similar to cancer, except, of course, in cancer, it occurs at a later developmental time point. So mosaicism is really just the occurrence of 2 or more genetically distinct populations of cells. This is really what it fundamentally is. And it can occur either in the same tissue or organism, any tissue that is actively proliferating, it can happen. And it does occur in almost every biological context. It's part of our normal development because these mutations occur all the time. It's part of aging, and it can occur as part of disease. So I will skip this for a second just because I'd like to kind of show you some case examples, and then we can go back to some of these, kind of interesting observations about it. But similar to PIK3CA, we found mosaic mutations of mTOR itself, the gene, in a broad spectrum of disorders as well. So this included focal malformations, where it's just FCD. And in these kids, the mutations were very low-level mosaic, which means that they were very restricted to the brain. Then we found some other children who had the mosaic mutation in variable distribution in both the body. So they had these linear hyperpigmentation in addition to the brain. And then we had kids who had actually germline mutations. The mutation was present everywhere. So these kids had, neurodevelopmental issues, a uniformly large head, no focal malformations for the most part. They had intellectual disabilities. They kind of looked like a lot of like P10. For some of you who might know P10 or Cowden syndrome, they just had universal abnormalities, but really nothing focal. And they had the mutation everywhere. So we see this broad spectrum across many of these genes. And this is actually a child who was at Seattle Children's, he was evaluated at Seattle Children's many years ago, and he was part of like this lovely piece on our website. So Aiden actually was a child that we had, seen and was followed by neurology and had really early onset epilepsy and had a focal malformation here that he had to undergo surgical resection for. And his family was generous enough to donate, whatever leftover brain tissue that was surgically removed, we were able to test genetically. And what we found was that he had an mTOR, a very common mTOR mutation, which is listed here, in his resected brain tissue. And the level of the mutation really ranged anywhere from 0% to the most anterior edge of the lesion to 9% at the core of the lesion. So there was this interesting mutational gradient within the brain tissue itself, within the same brain region. And that really kind of helped us understand further why his epilepsy occurred and why it required surgical resection. So what I've highlighted, hopefully, is that we have like this super fascinating and interesting kind of relationship between mosaic developmental brain disorders and the MTOR pathway and cancer biology. And, you know, so it's a really, really fascinating kind of relationship. And where do we, you know, so what is the value of this and where do we go from there? So this led our group to ask, well, many years ago, we asked two key questions, and we really dedicated all of our energies towards these two key questions. The first question was, well, can we reliably detect these? If they are brain specific, how can we diagnose kids with these? How can we, and importantly, can this be part of standard clinical care? Is this doable clinically outside of the realm of research and, you know, ambitious research, you know, avenues? The second is on the therapeutic side. If these disorders are truly due to mutations of this pathway, can we then treat these disorders using mTOR inhibitors? And so I'll show you briefly some of the work we've done on both of these in the last half of my talk. So in terms of the diagnostic side, when we started to think about this, you know, so there are three practical considerations to do this clinically, right? And these three practical considerations really apply to a lot of genetics. The first one is, what is the sequencing sensitivity? Meaning, are you choosing a sensitive enough method? Just like we talked about, you know, with an MRI, brain MRI scan being much more sensitive to detect a malformation than a CT scan. Similarly, in genetics, we've got to choose the right sensitivity, especially if a mutation is very tissue specific. That means that it's probably going to be in some cells, but not in others. So you need really highly sensitive methods to detect these low-level mosaic mutations. The second consideration is almost like a counterbalancing factor to the first one. It acts in opposition to it, which is that, well, how heterogeneous is this disorder? Is it caused by one gene or is it a lot of genes? And as I showed you from the mTOR pathway, you know, schematic, we have a lot of genes in this pathway. And a lot of the time, just by clinically looking at children and examining them, we really can't tell which gene is the cause just based on physical examination findings, because they can look very similar to each other. They can have focal malformations, some overgrowth, vascular lesions. It's a spectrum. So the more heterogeneous it is, meaning the more widely you have to look at the DNA, it really compromises your depth, so the sensitivity. And so that's kind of like these two factors are often in tension with each other. The third one is very practical, very straightforward. Most of us will get it, is, do you have the tissue? So for these disorders, for pediatric epilepsy surgical disorders, the ideal tissue would be brain tissue. And to be honest, had we not been really, I think, fortunate here at Seattle Children's by having an amazingly collaborative teams, both in neurology, neurosurgery, neuropathology, you need all of these, you know, people combined. It really takes a village in order to be able to clinically test lesional brain tissue that is surgically removed. It takes a lot of effort. And if you don't have that tissue, then are you out of luck? You know, then what? So that's really kind of these are the three important kind of considerations. So to really get at this, what we did a few years ago, and this is a paper that's now published in Brain from 2022, we had been collecting brain tissue samples as well as other biological samples from children with pediatric surgical epilepsy for many years. So we decided to do genetic testing on these samples. And so we took, you know, so we had 58 individuals in this particular experiment who had FCD or hemimeg. And we had about 124 total brain samples, including largely fresh frozen. And then we had also, because families are often very generous, and if they were willing as part of their enrollment in our research to donate other samples, easily accessible ones, hopefully, such as saliva or blood, we would, you know, accept them. And we tested these additional samples. On a small number of kids, we had some skin biopsy samples as well. But the vast majority were brain samples, as shown here in the black. So we decided to ask the question, can we pick up really low-level mosaic mutations in this cohort? So in order to do that, the diagnostic test we decided to use was something called droplet digital PCR. This is a newer PCR technology that really fractionates your PCR reaction into 20, ideally like thousands of droplets. So you get multiple readouts of the same DNA molecule. This is highly used in cancer now because it's very sensitive. So we decided to use this technology. We wanted to know how deep we can go. So we decided then we wanted to narrow our target so we can really go sequence deeply. So we designed what we call the MTOR hotspot panel. We took the 6 most common mutations seen in cancer and designed the probes and decided to test this cohort and these samples for these 6 mutations. And we derived something called the mosaicism level. What does that mean? Very simply speaking, we basically wanted to see what is the level of the mutation in those samples. So it's a very simple concept. We took the number of mutant droplets within our highly multiplexed PCR reaction and divided it over the total number of droplets we have. So that gives you kind of a proxy to the level of mosaicism in that sample. What did we find? So we found several really interesting observations. And sorry, this is a bit of a busy slide, but what I wanted to show you is 2 key things. The first one is if you look at this graph over here on the left-hand side, the x-axis shows some of the children, so de-identified numbers reflecting individuals who are tested. And then the y-axis is the level of the mutation as a percentage of cells in that sample. So what we found was that a lot of kids had mutations, but importantly, if you look, brain samples are in black here. So a lot of kids within the same child, sometimes we identified a range of mosaicism within the brain, ranging anywhere from 0% to sometimes up to 15%, within the same surgical field, if you will. And that was really valuable because a lot of other prior studies had only tested 1 sample per child. And when you do that, you kind of just get 1 value. So we were really lucky, again, that our team here is so awesome that we had multiple tissues from the same child. And so this was really helpful. And if you look here, blood is shown in pink. For many of these children, you don't see these mutations in blood because they're brain specific. So that's really, really valuable. And so if you look on the right hand of the slide, and this work, by the way, was led by a really talented research scientist who has since moved on to industry and is doing awesome things. So we came up with a few conclusions about testing, that are clinically relevant. The first one is that the range of mosaicism within the brain, even within the same hemisphere, can vary quite a bit. It can be anywhere from less than 1% all the way up to 20% as far as we know. It's possibly even wider than that, but this is to the extent of our knowledge. We also found that the level of the mutation in blood is almost always 0. There could be exceptions to this, but what this suggested to us is that blood is not an ideal sample, which is a problem because in clinical testing in general, we usually send blood from kids for a wide variety of tests, right? So this is an important consideration. If you look at other samples such as saliva, skin, then it's variable. Then it can be anything. So you gotta be careful. I think what we learned is that we have to be careful as geneticists in terms of which sample we're sending. We have to have a really thoughtful kind of plan for that. So I wanted to show you a few examples of cases to hopefully see how hopefully you can appreciate like really how relevant this could be and things like that. So this is a child who had been a long-standing patient at Seattle Children's who had a really significant hemispheric lesion, as you can see here. So one hemisphere was abnormal, dysplastic. She fulfilled kind of classic neuroimaging criteria of hemimegalencephaly. You can also appreciate that she had a vascular lesion on her leg. Not all kids with these disorders have these, but these are certainly very useful diagnostic clinical exam tools to kind of allow us to hone in on these disorders. So she had this vascular skin lesion. So we were able to perform diagnostic testing, and we sent several brain tissue samples from her resected lesion, as well as performed a biopsy on her skin from an area that was close to the vascular malformation. It wasn't right within the core of it because a lot of these can bleed, but it was close to the border. And we designed, as a result of our research, over many years, we designed a clinical panel at our CLIA lab at the University of Washington. So this is a panel providers can order, and we get actually requests for orders for this test. It's called the Megaplex. We get actually orders to test kids for this from across the country of late, which has been amazing. So providers actually have access to this testing and they can order it. And it includes a lot of the MTOR Pathway genes. So we performed the Megaplex panel. We found an AKT3 mutation on her that if you look here actually at the level of the mutation, it really ranged. But importantly, so in the brain, it was anywhere from 10 to 18% within the brain tissue. If you look at the skin, I really wanted to call this out because it was really low. It was only 1.3%, which means it's almost like it could be, if it was a less sensitive method, it might have actually been missed. And that's even while looking at a vascular skin lesion, not normal skin. So this really highlights, that how enriched these mutations can be in the brain tissue, and how tricky they can be, to find in non-brain tissue, which is a really significant complication here, because again, oftentimes we might not have access to testing the relevant tissue. So this is an example, one example I wanted to share. This is another example I thought I'd share as well, which is a child that was from out of state but consented in our research. And unfortunately, this child had really bilateral involvement, as you can see here. Bilateral, hemispheric lesion, severe cortical dysplasia, a really large head size, seizure onset very early on within the day of life 1. And he passed away within 10 weeks of life because sadly of his bilaterality, of his brain MRI abnormalities. And we only had actually tissue blocks from him. So we didn't have fresh frozen tissue, which really, I think, complicated our ability to test the tissue. Family was very generous, and they donated the brain tissue for, research and evaluation. And it was actually quite a large brain. It weighed, a lot. The expected weight at his age was about 500 grams, and his was like 900, and it was, severely abnormal and dysplastic. And we had only the ability to test a small number of them because the quality of the DNA, unfortunately, was quite degraded. So we ran into some technical difficulties, but we were finally able to do testing. And we found an AKT3 mutation, which is shown here, that was present in 38% of the cells. But interestingly, because we tested other, hotspots, other mutations, we found 2 additional, and these are shown here in these panels, 2 additional mutations at a much lower level in his brain tissue within the PIK3CA gene as well. And these are cancer mutations. So in total, we found potentially 3 mutations in his brain. One question we asked ourselves is whether or not the fact that he had, bilateral involvement that was quite severe and his epilepsy was quite severe, perhaps that might have been a clue that he has more than one thing going on, or more than one mutation, let's say, within the pathway. It is a possibility. We still don't understand the genotype-phenotype correlation very well within this spectrum. This is an ongoing kind of type of work we're doing, but it's just a hypothesis that, this is the largest number of mutations we have been able to find so far in one child, and perhaps it could explain the higher severity. I wanted to now really give kind of a quick word of caution, though, about all of this, so far, just as a quick kind of, pause here, that, I think a lot of these disorders, especially these brain malformations that are associated with early onset epilepsy, historically in the literature, they weren't really, targeted for genetic testing early on. We started doing this when I came to Seattle 10 years ago, and a lot of groups were starting to do it at the same time. But prior to that, the literature was always suggesting that perhaps they're non-genetic, they're random or sporadic. And so this is an example of a patient I wanted to share with you that was quite a learning experience for me as I was kind of doing this. Is, we had this child who was enrolled in our research from overseas, from Europe. She had a large head size, and she had hydrocephalus, actually. You could see here, the shunt marking. But we felt like maybe she fit on the spectrum because her brain was uniformly large, so she had megalencephaly, and we thought maybe she would fit. And at the time, unfortunately, we didn't have a lot of, there weren't any tissues to test. So we offered first targeted testing on her saliva, and we aimed for higher depth coverage. So this is what is meant by when we say 400 to 500x. It means just how deep is the coverage. So this is like really deep coverage to look for mosaicism. To compare it, exome sequencing is usually like 30 to 100. So this is like much deeper coverage, and it was negative. This was back in 2011. Then a year later, we said, all right, this family was very engaged. They really wanted to know the cause. We said, we'll do exome sequencing. Again, we didn't have tissues, so we tested blood and saliva on this child. We tried to do deeper coverage here, as shown here, also negative. Then in 2013, we had research whole genome sequencing accessible to us. So we did it on blood, and it was negative. So this was a bit of a, difficult journey for the family. They have emotionally been invested in this. They really wanted us to find the cause. We were doing a lot of testing. We were throwing, essentially the genetic textbook, if you will, on the 2 samples we had. And in a way, perhaps was that, the wisest thing we could have done versus like pausing and really trying to, talk to the family and meet them in a different spot of not just throwing one test after another. Because a couple of years later, the family consented finally to a skin biopsy on their child. And we did a deep exome. So we decided to abandon any targeted testing, and we just did an exome at the time. And their daughter had an MTOR mutation that was detectable in less than 10% of the skin cells. So she sadly passed away afterwards. Her condition was quite severe. She was neurodevelopmentally really delayed. And I think what the byproduct of all of this is that oftentimes, if we have the tools, but we're not testing the right tissue, it can be quite difficult for a lot of these families. And really, this diagnostic odyssey takes a toll. And it takes up a lot of resources as well. It could be better utilized to test more samples, more children, so this is just it was a lesson for me to kind of really have just a more cautious and just pause, at some point and kind of think about what we're doing and what is really the best path forward for testing these kids. This is highly complicated, and it's a very difficult journey to begin with. So we're set up here to, we're not always set up to succeed as providers, but it's just something that I think I reflect on. The last observation I wanted to show.
Dr. Mirzaa, I just wanted to. I'm echoing here a little bit, so hopefully it's not coming through. There was a question. So for the patient with AKT3, how would you counsel the family?
Yeah, great question. So there are several counseling aspects to that AKT3, for example, mutation. First would be on the diagnostic side, so we'll usually say this is a mutation that occurred later on during development, and but we always have to be careful. By later on, we mean later on within the first trimester of pregnancy, so it's still developmentally overall early, but not at the time of conception or not inherited from a parent, basically, and therefore is present in only a fraction of the child's cells or brain tissue. For a lot of families who want to have other children, this is very important because that means that the recurrence risk or the chance of having another affected child is extremely low. Of course, in genetics, we never say 0% because no pregnancy has a 0% risk, but we usually say it is as low as can be. It is less than 1%, it is not going to be passed on to another child. And that often gives families a lot of, relief from that concern. Then, of course, the other counseling aspects is the medical management. How does this impact the child's medical management? And I will talk about that in a few slides as it relates to like therapies and drug targets and stuff like that.
Thank you.
Yeah, absolutely. So this is actually, this is what this, is leading us to is, well, why is a molecular diagnosis important, right? So for many of us who have worked in this broad, range of biology here, right? A lot of clinical disorders are related to the MTOR pathway. This is not unique just to these malformations. Case in point would be tuberous sclerosis. Tuberous sclerosis is caused by mutations in TSC1 and 2, which are in the MTOR pathway. And if we've learned anything from the amazing wealth of literature on TSC is that the mechanisms of epilepsy, for example, in TSC are a multitude, and it's complex. So it can be growth, abnormalities. It can be abnormalities in the circuitry, abnormalities in the cells, or abnormalities in the molecules and how they communicate to each other. These are all just mechanisms of epileptogenesis in TSC from, again, decades of research. So the same thing really we'll need to kind of go through for mTOR-related, brain malformations. And importantly, because we have mTOR inhibitors, we have single inhibitors, where they target one node. We also have dual inhibitors. This is a very important question. So what I hope I've shown is that, before we kind of tackle therapeutics, we have to be aware of like the full picture and how, the layers of challenges we have, right? So we want to develop better therapeutics. Obviously, this is always an end goal, but in order to do that, we have to understand genotype-phenotype relationships. We have to understand tissue distribution, and importantly, accessibility is a barrier or, a consideration. We have to understand all the genes that are implicated because it's not just two genes like with TSC. We have a lot of genes involved here. We have to understand the level of mosaicism and as a result, the sequencing sensitivity. And then we have to understand, well, is it that the mutant cells are the ones that are causing the abnormalities or the clinical complications? Or could it be that the cells talk to each other? So there's something called, for those of any of you who've really done a lot of work in MTOR pathway, for example, there's something called non-cell autonomous effects, which means that it's not always the mutant cells that actually mediate the phenotype. Sometimes it's actually the non-mutant cells. These pathways, these cells talk to each other. And so it's possible that, you could have a mutation that's present in 1% of cells that actually causes widespread devastation because these cells communicate. So this is an important aspect that we still haven't deciphered because genetics doesn't address any of that, right? Genetics gives us an estimation of how much of the tissue is involved with the mutation, but it doesn't tell us functionally what is occurring. So really, to kind of address that, we kind of tried to develop a model that allowed us to kind of look at therapeutics. So we took samples, cell lines from children who were healthy, as well as children who had hemimeg or FCD, and using in vitro methods, using CRISPR-Cas9 or genome editing, we either introduced the mutation into the normal cell line or corrected the mutation in the mutant cell line, and we developed a wide range of neuronal cells in a dish. And I wanted to share with you here the two patients that really, their clinical phenotypes that we used for this model. The first one was a patient who had, or a child who had macrodactyly. You can see this overgrowth of his digits here, his fingers, and he had a common PIK3CA mutation. And then the other child actually had diffuse brain overgrowth, autism, epilepsy, intellectual disability, and she had an MTOR mutation. So we decided to take these two cell lines and start the experiments to really see what is happening at the cellular level. And I don't want to really spend too much time on this because this is like, a lot of nuance and work that actually was kind of impacted actually in part by COVID when the shutdown happened, unfortunately. But I just wanted to share some like high level findings here. But the two first questions we asked is, well, we know that these mutations are associated with brain overgrowth. Is it that we have too many cells, or are the cells themselves enlarged? So, the whole question of hyperplasia versus hypertrophy. And what we found is that there is actually a little bit of both occurring in these, disorders. We found that if we looked at cellular or population doubling time, so this is a reflection of how fast the cells are dividing. If we looked at the PIK3CA and mTOR mutant lines, they were dividing at a much faster rate than normal cell lines. So there was a larger number of them. So there was some element of hyperplasia. Similarly, if we looked at the cell size, including both the nucleus and the cytoplasm, we found that these mutant lines shown here, PIK3CA and mTOR, they were larger, both in the nuclear and cytoplasmic compartments. So that was really helpful. So there's an element of both hyperplasia and hypertrophy. And if, for P10, for example, this similar findings have been seen as well in some of the cancer, of course, disorders. Then we developed what's called the brain in a dish, model, which is what's called also organoids. So where we take these cells and we develop them into like this 3D, model of brain tissue. So we allow them to self-organize. And I hope what you can see here is that if you look under this is just under the microscope, it's almost under the naked eye, really. The PIK3CA and mTOR mutant organoids were much larger, reflecting or recapitulating the actual human phenotype. And they were much larger than the controls. And even the mTOR one looks quite dysplastic and abnormal, and there's something really, wrong with these cells. So that was really, really helpful. So it recapitulated in a dish the actual brain phenotype. So then the other question came to therapies, and why is this even important to begin with? For decades, a lot of these kids would go through kind of a same, therapeutic kind of journey with variations depending on the group and the center about which, therapeutic to prioritize first. But essentially, a lot of the kids would first undergo, anti-seizure medication trials, right? So they would be treated by a wide variety of anti-seizure meds. And of course, anti-seizure meds, there's so much improvement in the number of them and their side effect profile and things like that. So those have changed over time. But anti-seizure medications are usually tried. Sometimes the ketogenic diet might be tried. And then afterwards, if epilepsy continues to be intractable, many of these kids then undergo epilepsy surgery. And sadly, as much as epilepsy surgery can be life-saving in many of these children, for some kids, epilepsy recurs even after surgery. So this is where this question that I'm posing here becomes relevant, is then, knowing that some of these disorders are caused by mutations of the MTOR pathway, could we use mTOR inhibitors, just like we do for TSC, to treat these disorders? So on the cell lines that I showed you, we decided then to use 3 inhibitors. Rapamycin is the famous mTOR inhibitor, which targets mTOR. We used an AKT inhibitor, which is experimental, called AKT inhibitor 4. And then we used a PIK3CA inhibitor called alpelisib. Alpelisib, again, is actually a newly FDA-approved inhibitor that is used to treat PIK3CA positive breast cancer in women. And it also recently became approved for PIK3CA positive body overgrowth and vascular malformations. The problem with PIK3CA, with alpelisib, is that the brain brain penetration is not very well studied. The PKPD data, pharmacokinetic, pharmacodynamic kind of information about its effects on the central nervous system is limited. So we thought, why not? Let's just use it on our neuronal cell lines. And this is the last data slide, I promise. But we essentially found that when we used rapamycin on the 2 mutant lines that I showed you, it actually rescued the pathway hyperactivation that we saw in these cells. This is actually almost a proof of concept because this has been known before from the TSC literature that this is why rapamycin is used, is that it helps rescue mTOR hyperactivation. Interestingly, for the newer drug, alpelisib, which again is becoming more widely, oops, sorry about that. Oh my gosh, I don't know why this panel kind of showed here. Alpelisib, actually, what we saw is that it actually rescued the hypertrophy. So the cells, after treatment for a while, they became smaller in size. These experiments are completely preliminary, and this is when the shutdown really impacted us. So we actually haven't had the opportunity to go back and continue some of these experiments, but it raises the promising question that perhaps we can use, or we can think about using some of these mTOR inhibitors for kids who have epilepsy after surgery. If they continue to have epilepsy, we're kind of then running out of options there. So perhaps mTOR inhibitors could be a promising avenue. So with that, I will really wrap up and say, where are we now? So here at Seattle Children's, where we are with all of this right now is that, we've got all these diagnostic tools, particularly the NGS tools. In the era of genomics, for pediatric epilepsy surgical disorders, we have another paradigm, which is what we call tissue-specific genomics, which really requires us to have ultra-deep sequencing and understand brain-specific mosaicism and brain-specific, genetic abnormalities. And luckily, here at Seattle Children's, we have really the opportunity to implement, and we've built all of this into our clinical care right now. So, we see these kids in the epilepsy genetics clinic. We offer them pretest counseling, we offer genomic testing, and then we, counsel them and support them after the test is done. And we have the megaplex panel that I showed you, at the University of Washington to order. And the hope is that with time, if we have candidate drugs, we can enroll them either in relevant trials or offer them an appropriate medication if needed, and if surgery is not curative in that sense. So, and this is all really part of just to take us finally a step back from all of this. This is part of a broad, wheel of things that have to occur in order for us to be able to do all of this, right? So when we think about all of these disorders, it really takes an extremely multidisciplinary group of people to really be able to do all of this. And I just kind of shone a light during this talk at probably one aspect of it, which is just the diagnostic kind of arm. There's a lot of amazing work that's being done by our colleagues at Seattle Children's and elsewhere on understanding the natural history of these disorders. We need to identify what are the therapeutic targets. Is it just seizures or is it other comorbidities, right? There's a lot of work happening at SCRI and other centers as well on the preclinical side with animal models and other cellular models. And then lastly, this is where we're hoping to go with our newly launched neurotherapeutics program, is then really kind of how do we efficiently translate all of this into the therapeutic side and offer families the right support for clinical trials or other, other avenues. And so these are all kind of multifaceted approaches and we really need all of these to kind of happen in tandem in order for us to really kind of make progress. So with that, I just wanted to first thank you for spending the evening listening to me talk about these disorders that are near and dear to our heart. And this is actually, again, we're just one of many champions that really did amazing work, both in neurology, surgery, neurosurgery, neuropathology, lab medicine, and my lab at the time working on this and our, funding sources. And with that, I will say thank you so much. And hope this was useful. And here's a nice picture of Rainier in the fall from last year. So I welcome questions and comments.
Yeah, please feel free. You can put it in the chat, or I think your mic should be unmuted, and you can feel free to just ask a question. All right, well, I'm not anybody. That was excellent. You were right, a perfect 55 minutes. So that was great. Okay, so I'm still looking at the chat box if anybody wants to throw a question in really quick. And I did want to remind everybody that we will be following up with an evaluation link. So please just take a couple of minutes and fill it out. We would really appreciate it.
Hi, this is, I'm not sure if you can hear me.
Yes, we can.
Okay, took some time to find the unmute button, sorry. Kamadin Klein from Calgary. Just wondering, so the testing for somatic mutations that you presented, so first, very interesting talk and great topic. So the testing you're doing, is it actually covered in the healthcare system, or is that more on the research side? And I understand you have kind of integrated the research, of course, with the clinical, but how close do you see this to the actual clinical use that it's covered by the healthcare system?
Yeah, that's a great question. So, and this is actually something we're figuring out as we go right now. So, we've started to actually order this testing clinically. So, meaning we're asking insurance basically to cover the cost of testing. The premise of it, the core concept is that it's medically necessary. So as long as we feel like it's medically helpful to the child, I think this is really the key thing. If it medically helps them, then my view, at least in my humble opinion, as a provider, is that then we should do the test clinically and not under research, because sadly, at least in the United States, our research laws is that any test done in the research lab, like my lab at the time, it can't go into the child's medical record. It cannot be used for, depending on the research protocol, obviously, there's a lot of differences, but for our protocol, for example, we couldn't issue a report that goes into the child's medical record. It could not be used for future, pregnancy testing, recurrence risk, counseling and family testing. It had to be confirmed clinically. So even after we did the research, we still had to do the test clinically. So Europe has a different set of rules, I'm pretty sure, with all of this. But so as long as the test is medically necessary and has implications for medical management of the child, then I'm hopeful that we would be able to kind of get it integrated into their clinical care. We started to do testing clinically last year, and we so far have been approved most of the time to do it clinically. So, and that's been helpful. I hope that kind of answers the question, but I really personally think that this should be part of their clinical care. Just like in cancer, we want to find the mutation. For epilepsy, we should look for these mutations. I think it can be helpful.
Thank you very much.
I have a couple other questions. So I'll ask the first, you can answer it, and then I'll ask the second, and we'll see if we get some more. But why do the genes triggered to present this way?
Does the question mean, I'm curious, does it mean why do these mutate, why are these genes mutated to begin with? Why is that, or why are they?
I read it verbatim. So why do the genes triggered to present this way?
I guess my question is why does this happen? Why do these genes get turned on?
Yeah, that's it. Yeah. It is. Thanks for that clarification. It's interesting, right? Because a lot of these mutations are recurrent. So what we call hotspots, meaning the same amino acid substitution can happen over and over again, just like in cancer. So there has to be something about the DNA qualities at those regions that make them more prone to being mutated. We don't know. It's not a DNA fragility issue. It's not, that there's some damage repair response issue or something like that with the DNA. But for the most part, these all arise postzygotically and spontaneously, So we really, honestly, I don't think we really know. What we do know is that there are these hotspots within these genes. But yeah, for the most part, they are spontaneous. Something that, while the cells are dividing, something triggers these mutations to occur. It's not environmental as far as we're aware of some type of damage or, insult to the cell, as far as we know. So radiation or things like that. We're not really like, there's no data to support it. But yeah, it's a great question. We still don't know. It's the same mutations like that, again, are usually implicated in cancers in adult form. Same mutations we see over and over again. You know, like the AKT3 mutation that I showed actually is the most common oncogenic mutation in AKT3. It's at E17K, at the amino acid, the 17th one. Fascinating, but we really don't understand it very well. And I think I see, Patty, another question.
Oh, yeah. Yep. So they're wondering if you have any thoughts about, are you reading it or do you want me to read it? Oh, well, I should read it out loud so everybody hears it. So if you have any thoughts about why, and I'm probably going to say this wrong, but vigabatrin is perhaps the most effective and prevents some severe epilepsy development in some patients compared with the natural history in patients with TSC particularly.
Yeah, that's a great question. To be honest, I don't know why vigabatrin is much, you know, one of my more esteemed neurologists would have, you know, a wealth of, like, there's a lot of, like, I think, literature showing the efficacy of vigabatrin for treating TSC. And then I know rapamycin, there was a trial, of course, for looking at the efficacy of rapamycin. I am not experienced, I'm not a neurologist, so I will say I don't treat my kids with vigabatrin. So, yeah, it's a good question. I'm not sure. For these disorders, I don't think for MTOR, you know, related brain malformations, I'm not aware of systematic literature, to my knowledge, that says that vigabatrin is as good as it is in TSC. It's possible that I could be missing that or something, but yeah, it's a very interesting question. The other question actually we've been wanting to ask, or we've been asking, is also the ketogenic diet, because there is some literature, of course, that the ketogenic diet has an impact on the MTOR pathway, and it can suppress the pathway. There's been animal models that show this. So the question would be, is then could the ketogenic diet, you know, be a molecularly targeted therapy for these kids in a way? That's another interesting question we've been thinking about. But yeah, sorry, I don't have a satisfactory answer on the vigabatrin front. Welcome.
Well, they thank you anyhow.
Thank you.
I forgot to ask you at the beginning, are you going to be at the symposium in October speaking again this year? Yeah, so everyone, and you'll get notice, but in October 14th, they're going to be doing the second annual epilepsy symposium. And so Dr. Mirzaa will be presenting more, as well as several of the other colleagues specific on epilepsy. So definitely look for that. And I don't see any more questions in the chat, and nobody's speaking up. So thank you very much. And thank you, everybody, for joining us this evening. Oh, wait. Nope, that's it.
Thanks, everyone. Have a lovely evening.
Thank you. Bye.